So Dr. Feng he got his Ph.D. in 2011 in stony growth with Dr. Lincoln. And you guys have probably heard of Lincoln. He was one of the pioneers of the field, and really one of the early people to start doing bioinformatics. So it was very good training that Dr. Feng got. And actually, I was just talking to him earlier, and what he was telling me is that in order to get into Dr. Stein's lab, what Dr. Stein did was send him an encoded email message and then ask him to respond to it. So then, so Xin Feng had to go back and figure out how to decode this message and to give Lincoln Stein the appropriate response, which he obviously did a really good job at. And then after he joined Dr. Lincoln Stein's lab, he did very, very well, published 2 science papers, and got out quickly with his Ph.D. Since then, he was recruited to the University of Texas. Go ahead. Okay, the University of Texas Southwestern Medical Center, where he worked with he was recruited by Dr. Helen Hobbs to set up the next generation sequencing pipeline, and there he developed a lot of software to analyze next generation sequencing data. And then from there, he also worked at next door in the Genomic College of Medicine in the Human Genome Sequencing Center. Throughout all this time, he's done very, very well, published a lot of very high-impact papers, including the papers that I talked about, and also developed new methods for analyzing chip-seq data that is arguably one of the top methods in the open space. So this talk today is titled Towards Precision Medicine, Advances in Informatics for Genome Variance Analysis, and I can't wait to hear what he says.
I would like to thank Crowdsourcing for inviting me to this very exciting talk, and also thank Dr. Chan for his wonderful introduction. Actually, I think today what I'm going to talk about is actually more on our user-centered goal. So I hope that this will be a topic that we can share with each other about our each other's ideas and really on this very exciting topic.
Okay, I'll just unmute my microphone. So actually, I prepared some notes along the side, but I think today I cannot see the notes because I already have a presenter view. But as you can see from the title, so what I'm going to talk today is about the very huge topic. Is called precision medicine. So it's proposed by President Obama back in to the start of 2015. And today, what I'm going to focus is a very particular aspect of this initiative, which is on genomic variant analysis. And in more details, I will be talking about our efforts in better trying to advance the methods in a particular field. So the first thing is, what is really about the precision medicine? What can every people get from this initiative? I think from my personal perspective, this PMI is more about getting better healthcare for patients and also providing more opportunities for researchers to do some research at the intersections of a lot of different fields, such as genetics, health data, which is more like EHR, and also even the ecology and all these related public health topics. So the goal is that we want to bring forward the care that we can give to our patients to get and so that we can get everybody a better and healthier future. So in terms of data, I mean, if you view the PMI from the perspective of data, this is what I really like. I mean, so basically, if you think a person you can think him as a particular genotype, and the genotype interacts with the environment, and the environment plus the genotype will produce some phenotypes, which like, are your hair curly? Do you have red hair? Or do you have something else that other people don't have? And from this interface, we can generate a lot of data, different types of data, such as genomic data, the gene expression data, the proteomics data, and we also have some patient-generated data, such as, for example, if you wear some Fitbit devices, they can collect your pulse, your blood pressure, even the miles you travel every day. So I think this is a very good opportunity for School of Biomedical Informatics, as well as the new Center for Precision Medicine, because this really is a collaboration between health informatics as well as bioinformatics, which is what I'm coming from. So you may ask why we should do this right now. I mean, the idea is very good, and I believe that many people have been thinking about this. I mean, a better, more personalized medicine, customized to each individual patients. And we can also integrate different types of data, as I mentioned in the previous slide. Then the question is, why should we do it right now? I think, according to these official slides, the biggest factor now, the contributing factor or the pushing factor is the readily available human genomes. Basically, now you can get your genome sequence in much, much cheaper and much, much more accessible ways compared to almost 20 years ago. And also, we have much better improved technologies for biomedical analysis. And I guess every people has probably heard about Amazon. And Amazon has been famous for its online web store as well as its cloud services. So these tools have been enabling us to manipulate a very huge volume of data sets at a scale that is unprecedented. So I will give you some concrete examples to demonstrate the three reasons. This is a snapshot from NHGRI, and I think if you take a look at this one, originally you had to pay almost $100 million just to sequence one genome, and this is where my Ph.D. advisor Lincoln Stein was working with. So he was basically in the very pioneer stage of this human genome project, and what he did is to design a browser for the genome data for probably just one person, one reference genome, basically. Excuse me. But nowadays, as you can see, starting from around like 2008, 2009, where the Illumina really stand out, the process for sequencing a whole genome really drops out significantly. And all the way until maybe the end of 2015, where I think there is a famous genomics company called Variance Genomics, which is actually based on Boston. They announced that they can do the whole exome sequencing or whole genome sequencing at a very unprecedented price. And specifically, they mentioned that they can do this for only $1,000 for any person who has this access to this technology. So compared to just less than 20 years ago, the drop is significantly. At the $100 million, that means the technology is not even for rich, it's for super rich. But today, the technology is for everyone. So every patient can have the chance to enjoy a better patient care just based on your personal genome data. Another example I want to mention is actually this small smart device called Flash, because this device has been so popular that there has been some opportunities centered around this device, and also as well as the huge volume of data collected by this device, like your biometrics that is collected 24 hours, 24/7, and almost all year round, as long as you wear this device. This device can provide something that is very important for the doctors, for diagnostic centers, and for the labs to make some decisions. So it will be very interesting that we can just utilize these new devices as well as the data that they can provide to do some very exciting research at the intersection of different areas. And you can get this for, I think, less than $100. So it's really accessible to everyone. So given all this new exciting news, I think especially after President Obama announced his PMI, NIH has decided that they will put, actually it's not NIH, I think it's something above NIH, decided to put around more than $200 million in 2016 just to support this initiative. And the initial plan is NIH will get more than 70% of this money, and the rest, and the majority of the rest will go to the NCI, which is more on the cancer side. And also, there are some other institutes that can get some money from this big initiative. So it's a lot of money, which means a lot of opportunities. That's from the view of academia. If you take the view from external side, I mean external worlds like venture capitals, the news is also very good, because just right after the announcement of PMI, during the first quarter of 2015, the top 10 investments, which in total is more than $400 million, there is about 1/4, which is around like 25%, is in genomics. So that's, even this part is another $100 million. And now today is 2016, the second quarter. So if you think about all the attractions this PMI can attract, then you can really think about this a huge pool of money that is, and there are new money that keeps flowing into this pool, which means a lot of opportunities for us. Do I have a, okay. So as I mentioned at the very beginning, there are 3 factors, but today I'm going to focus on the genome part. So I think everyone knows this very famous talk. It says that almost any 2 people is almost exactly the same, except for that tiny bit of difference in their genomes. But actually, this tiny bit of difference, I mean, just by looking at this picture, you probably cannot feel how tiny they are. But if you realize the difference in comparison to our genes and the whole genome, you probably can see this little tiny part is really that significant. However, this is a very counterintuitive, because, for example, the most majority of this 0.5 difference are composed of SNPs. What are SNPs? SNPs are the tiny bit difference among your genome. For example, if you compare you and me, we probably are the same for most of the parts in our genome, but at that 0.5% or up to 1% size, we are different. You are an A, I'm a G. You may consider this one base pair difference not that significant, but there has been many studies saying that with just one base pair difference, you can get totally different phenotypes. For example, your hair is curly, but my hair is tight. And your hair is red, my hair is black. So this thing is very important. The thing is, we now have almost 150 million SNPs already reported to DBSNP, which is the central archive of all the known public SNPs. And we have also 20,000 genes, maybe even more than that, to discover. The thing is, for most of these things, the 0.5 difference, we knew. But today, I think we can still say that we know little about these things. So actually, I took this picture, I mean, this background image, when I was in American Society of Human Genetics meeting in 2014. So I was presenting something that I'm going to talk about later. But I'm really thinking, I mean, I was really thinking at that time that we should do something. We should do something to promote our understanding of this huge cohort of SNPs. And we hope that by doing this, we can really advance the human healthcare. So that's why we come out with this ClinGen project. This ClinGen project is short for Clinical Genome Resource. It's a $50 million, 5-year span project, federal project that is sponsored by NIH. So we involved, I think, at least 3 major institutes from different parts with different specialties and expertise. We also have very diversified working groups, a panel of working groups who have people major in all different kind of areas. For example, in my part, we have Dr. Carlos Bustamante that is chairing the informatics part, and we also have the CLAMR database, which I will explain later. The CLAMR database part person, we have Melissa Landrum, Donna Magalot, who have direct control of this very precise human disease database. So the goal of this project is to somehow increase or raise the quality of patient care by doing this different type of things. So the first important thing is we want to standardize in the annotation and interpretation of genomic variance, which is the 150 million stuff I just mentioned. We also want to have a centralized data house for all this data so that our clinicians, our scientists can access this data whenever they want. We also want to somehow get some help or leverage the existing developments in machine learning to somehow automate this annotation process so that we can improve our throughput, our efficiency of variant annotation and interpretation. We also want to have somehow build an evidence-based process for curating these variants and genes. And lastly, we also want to somehow help the people to decide if some genes are medically actionable or not. Excuse me. So I will be talking about some of the points in our later slides. So this is the organization map for the whole ClinGen project. As you can see, it's actually centered around patient and family. So right outside the patient center, you can see there is this EHR ecosystem, which somehow collects and stores and manages the patient-generated data, such as pulse, weights, body temperature, something like that. And here comes this ClinVar database. The ClinVar database somehow will be using as a resource to support this EHR system so that we can get better healthcare to our patients. And this is where I was. This ClinGen DB is a much larger cycle. It covers a much larger area, and it talks to different parties. It's probably the component that talks to the most parties in this organizational system. As you can see, it talks to our expert working groups. It talks to our machine learning algorithms. It also talking to external data sources. It also has to somehow be responsible for the data down from ClinVar so that it keeps improving the database itself. And also it has to push back the data and conclusion from the working groups as well as external databases back into ClinVar so that the patient can directly benefit from ClinVar through the EHR ecosystem. So I was actually at the center of this ClinGen DB stuff. So in the following slides, I'll be talking about what we did with this ClinGen DB, how we designed that, how we populated this database initially, and how we benefit from the data, and what's the preliminary findings we got by playing with this database. So actually just yesterday, actually last night, I got the news that FDR just issued a draft guidance for using public database to support the annotation and interpretation of the genomic variants. This is actually right on, it said July 8th, but it's actually on July the 7th. That means that I think FDA and almost every party has been on board, and the same boat, to say that to have something like the ClinVar or ClinGen DB is vital, is essential to the success of precision medicine. So getting back to ClinGen DB, this is our initial draft design of the whole data store. So basically, we start with a variant identity index, basically like chromosome 1, maybe in gene PCSK9, and in a particular location. We use that to index all the information in our database. And we use this index to correlate with our external databases, such as ClinVar, and more than 100 different types of locus-specific database, such as a database just for the gene TP53 or just for the gene BRCA1 or A2. And we also have to deal with all the different types of population data, especially their frequencies. And also some protein features, protein structure features that can be useful for their machine learning algorithm. So the idea is once we bring in these different types of raw data sources, we can do some operations, we can manipulate these different types of data sets so we can have a derived data collection for different purposes. For example, if, as I mentioned, we can probably use the protein structure data to do some machine learning. So the machine learning algorithm may be something like these apps. So these apps can use the APIs, the RESTful APIs, to directly query our database without talking to us. So in that way, the user of this database can generate the data just based on their demands. They don't have to talk to us. And we can also do these things ourselves. For example, we can integrate the ontology data or other types of structured types of data, such as semantic webs, RDF stores, or other things, to somehow keep enhancing these data collections so that we can produce more things that our apps find useful. So if you view the whole process from the view of data or information flow, it's actually more like this. So initially, we have all different types of data pipelines that can clean, enrich, and annotate all types of different data, such as the famous One-Song Genome Project data, or other types of database for a particular set of genes. And then we bring in this data, and they come in as individual data collections. And then we talk to our domain experts or our users who have specialized knowledge or expertise on a particular set of genes or domain questions. And they can push back their knowledge into our raw data collections so that we can get some aggregated data collections. And this aggregated data collections can be further enhanced by our variant curation UI, which can further abstract the knowledge from our variant curators. And this curator also curate and review the data. And once they finish this process, this data can be either pushed back to ClinVar, which is the so-called central archive, or it can be consumed by other types of apps.
So like this flow chart,
yeah.
How the different groups contribute to this work together? So like pipeline, curation,
yeah.
and storage and so.
Exactly. Yes. Yeah. So to be simple, I think most of the data stay on our side. We basically we're like a sponge. We taking all the data, but we have to somehow provide interfaces for both computers and curators. For the curators, we provide this UI. We also have some informal meetings with our domain experts. For example, the people from North Carolina, and they provide us with either Excel sheets, which we somehow go back to the data pipelines to push them in, or they directly use our web-based user interface to enter this information by themselves. I'll be talking more about this on later stages. And when the data is in a database, as I said, the data can be accessed via API. So the people who want to use our data don't have to talk to us. They just use the predefined APIs, basically like a link, to directly access the data that they have the accessibility.
So who wrote the API or developed the API?
I did.
You did?
Yeah.
So roughly which kind of structures?
The API is actually not the most difficult part in this thing, because our data is more like a tabular data, which means the data can be accessed in a very limited ways. So that way you don't have to design too many access points. You only have to design, for example, reading, writing, deleting, and posting. And actually only a few people can deleting or posting. Most of people can just read, and that means we don't have to spend too much time on the API part. The most difficult part is actually this cleaning process. You have to consolidate different parts of the database so that they talk in the same language, so they can all be pushed into our data stores.
So did you develop the standardized
yes.
procedures?
I'll be talking about that later. Yeah.
So maybe one more question
Yeah.
here. This is maybe a little later stage.
Yeah.
So it's very often, the same data.
Yeah.
The same data means same variant. People have different.
Yeah.
Something should be clinically relevant, some not, something related.
Yeah.
How, in the experience in clinical, how did you?
I'll be talking about that later. I'm glad that we have so much in common. So, excuse me. So let's go back to this organization chart. If you take a notice at this, you can see that ClinVar has to be synchronized to our ClinGenDB regularly. That means you really have to know how to get the data from ClinVar. And as Dr. Zhao just mentioned, we are not just like data staging services. We have to take in the data, but at the same time, we have to also, provide our data to our external collaborators, like as Dr. Zhao mentioned, like physicians or clinicians who want the clinical part of data, or we had to provide the data to people like me, bioinformaticians, who are more interested in the overview, the big picture, the identity of the variants. So how do we do that? Basically, the thing is you have a you have a single bottle of water, but you want to use the water in many different ways. Then how do we do these things in a very efficient way? So okay, the claimer stuff, why it is so important, I have to give you some backgrounds. In our whole design, the claimer is the receiving point from 4 different stakeholders. We have to get the data from researchers. We also have to absorb the data from patients, as well as clinics and all other labs who have their own data silos. We want to bring all the data into claimer as much as we can. So why is claimer so important? So this is the official website of claimer. There are so many things on this website, but I think if you take a notice at this, there's a clinical significance that can say that's the answer to this question, because this is probably the only database at this moment that has this authority and credit to tell you if a variant is pathogenic or not. Only this database can do this at this moment, so it's very, very important. But if you go to the original data of claimer, you find that reading or understanding this data is non-trivial. So the most important part of this claimer database comes in as a special file format called XML. It's kind of ancient, but it's something that is very popular in the field of computer science. But for most regular people like us, this is not what we want in front. And even for this XML file, if you want something, for example, take the genomic location of this variant as an example, it has 2 different assembly, 37 and 38, which is actually 19 and 20. If some people are more familiar with that, you see that it has 2 different locations. So, for example, if you want the data, you tell me, I want data from claimer. Then I will ask you, which location do you want? Then the people probably don't even know what I'm talking about. So in that sense, I probably would just give him this one as a default. But then maybe later Dr. Zhao come to me and say, Xin, we are updating our pipelines, and I want the latest coordinates in maybe HG20. Then I have to go back to rewrite my program completely. And that's a very time-consuming process, and it's also very error-prone. So to solve this problem, we basically want to say that this is our goal of the design. We want the specification to be independent of our software. So whenever the specification change, you only change the specification. You don't go back to change your software. So this is what we come out. We basically come out with a specification. We call it the map. Basically, this map, for example, this one says, if you want to know the title of this SNP, you go to this particular XML path. You tell the program that, OK, you go to this place, and you get the title of this record. You want to know if this variant is pathogenic or not, you go to this place. And this place will tell you if it is significant, if it is pathogenic, and when is that evaluated, and what's the current status of this evaluation. You can do all these things just by writing this specification. So that means when your demand is changing, say you want something, OK, I don't want this assertion type anymore, then you just delete this row completely. And you don't have to go back to rewrite your program. So, and this thing will directly translate the XML file into something that is more human readable. And for some of us, probably know the JSON file, and this is a JSON file. It's much, much more human friendly compared to XML. And we also have the convenience to redo the specification. I mean, basically, anytime you want. And this software is now readily available at the GitHub repository, and it's very easy to run. Basically, you just download the codes, do some very easy setup, and you can just use one line to run the thing, and you get whatever you want. You can run that against one single XML file, which means one variant. You can also run it against the whole claimer database dump. It's very memory efficient, so you can run it even on your own laptop.
So I'm not very familiar with the JSON pipeline system. How popular it is in the biomedical research?
It's probably not that popular in the field of biomedical informatics, but it's the state of art, the standard, the de facto standard in the field of I would say, modern internet-based software engineering. So it's the standard. Yeah. And actually, we need a JSON to make sure that our API is working efficiently. Yeah. OK, so I was talking about how we basically get the data into our database. Then the one way we did is we consolidate the database ourselves. Another way, which I'm going to talk about next, is to enable the curators, the stakeholders, to contribute their knowledge into our database. So we somehow defer our burdens and our responsibilities to people who have much better knowledge than us to do this. So talking about curators, I'm giving some background information about how you actually curate one particular piece of gene. So what you do is you first choose the gene you want to curate. And you then pick the disease or the condition you are interested in, for example, Lynch syndrome or breast cancer. And then you do some search in PubMed about like which papers are talking about this particular gene disease pair. And then you get their PubMed IDs, and you get their other information such as authors, years, or major findings. And then you continue to the rest part, like what's the variance mentioned in this paper or something like that, what's the conclusion for this variance. So this is what we did in our original UI. So now I'm going to talk about how we help these curators to get their performance to up to 250% gains. So the first thing we did is whenever the user enters the name, excuse me, we automatically find the official name for that disease from two popular disease ontologies. One is ALMIM, another one is the alternate rare disease ontology. So the example is, for example, what I mentioned is Lynch syndrome. You just type LYN, the first three letters, and the user interface will automatically pop up this thing for you. And this thing is not from us. This thing is where the API from, I think this one is from ALMIM. It also provides you with all the synonym that you can consider if you are not thinking about Lynch syndrome. The second thing we did is about the search. So what you do is typically you type in the gene and disease name, and you hit enter, and you analyze all the things by yourself. So what we did is we do the search for you in the background whenever after you give us the gene disease pair, and we also save your query back into the database just in case you want to reperform the query later. So this is an example. So this is the results after typing the gene and disease name from PubMed, and this is the search details. We save these search details into this query term, and we automatically give you the most important information about these publications back into the UI, so you don't have to do this. So typically, if you do this by yourself, by hand, you probably spend like 10 minutes. But if you use our UI, this is just one click away.
What is the UI? Is that software that you
develop? Yeah, that we develop. Yeah. We developed for this project.
Okay.
Yeah. So the third thing we did is this is the most tedious part in this whole curation process. So basically, once you get the publication, you have to go look into the details of each publication and you search for the names, years, institutions, and all the things, because these things are considered as provenance of this search. So you have to keep that. They're very important. For this part, unfortunately, it's the most easiest part for the software because we can use the API to automatically extract all this information for you. You don't have to do anything about this. The last thing we did is actually, I think, more meaningful for the whole process. So one important thing for the curator to do during the whole process is to actually read through the whole papers and extract all the variants mentioned by the paper. For example, for the BRAC A1, you probably go to RSAAA 489 or something like that. In order to do this, you have to manually scan the whole test. I know there are some software doing this. So what I was thinking about is probably we can do this by leveraging some existing software. So what we did is to use a software called TMWAR, which is developed by a team in NCBI. They probably come out with this a few years ago. So we can just so whenever the paper with the PubMed ID is entered into our UI, we can run this at the background. So it automatically extracts all this variant mentioned in the abstract of this paper and fill that for you. So everything is done automatically. You don't have to do this by yourself. So with just these 4 different steps of like intuitive steps, we can see that the whole process is now greatly improved with our UI-based assisting process. And actually, we see that after we talk to our users, we see more, up to like more than almost 3-fold of performance gains in terms of the curation time you need. So let's go back to these goals of ClinGen. Another very important goal of this project is to actually implement evidence-based expert consensus process. What does this mean? This means that we had to come out with a guideline, like what FDA does for the database stuff yesterday. We have to come out with something like a guideline. So everybody in this field who wants to do something like this, who want to contribute to this database, has to follow this process. So again, we make sure that we talk in the same language. So then Heidi Ram, the leader of this ClinGen project, came out with this paper which describes the first draft standards and guidelines for how to actually report this variance using a rule-based process. And SMG stands for American College of Medical Genetics and Genomics. It's our probably the most important society of our field. So the most important part for that paper, that more than 15 pages paper, is actually this matrix. So this matrix may be very complicated to read initially, but if you see on the top, basically it says that this 3 columns says that this evidence can prove that this variant is benign, which means it is not harmful to human bodies. And this 3 columns of data, if you have anything falling in these cells, that means this variant is probably pathogenic. So, for example, if now I have a variant, I want to follow this process, what do? Then I start with, for example, the first cell. The first cell says that you have to first look for the minor allele frequency for this particular variant. So if this variant is, say, 40%, which means quite common in the population, that means this variant is probably not harmful at all. So by going through just the first cell, you can stamp the variant says it's benign. But in most cases, things are not that easier. For many situations, you have to go through almost all the cells of this matrix to decide if this variant is pathogenic or not. And what makes the things even more complicated is that in order to have the conclusion, to have the stamp, whether, on the pathogenicity of this variant, you actually have to satisfy more than one condition. For example, if you want to see the variant is pathogenic, you have to have at least one very strong evidence. Plus, you have to at least one strong evidence, or you have more than 2 moderate evidence, or you have more than 2 supporting evidence plus something moderate and something in between. So this thing is, this thing is very complicated, and it's particularly complicated for human to follow. So what we come out is that we want to have something that somehow implement this whole process. So we free your mind. You just focus on curating. You just focus on providing information. And we help you with the whole process. So you don't have to worry about like, oh, is this variant pathogenic or not by going through this whole process. And another very important thing for doing this project is a lot of clinical labs, diagnostic labs are also trying to, actually I think more or less they're forced to follow this rule because this is the future. This is guideline that everybody has to follow. So they have to evaluate how will this guideline help us? How much efforts it will take for us to adapt to this new guideline? How much time, how much human resource, how much change I need to take? Yeah, we welcome changes, but I'm not the first. You are the first. That's what people are thinking, most of the time. So if we can come out with a simple implementation of this guideline, of this rule, then people may have a very concrete feeling about the cost, about the time and the efforts that it will take for them to make the shift. So that's why we come out with this web-based apps. I probably will go out, jump out, and to give you a live demo. So this is the real website. So if you go to the website, the first thing you can do is you click here to give your variant a name. For example, I can give you a test variant. for demo. And now the most important thing for you to know about this variant is pathogenicity. And currently it says not enough evidence, which means you have to give some input to the system so that the system can make the evaluation for you. As an example I give in the very beginning, the first cell is probably the most easiest part. You can just provide the population allele frequency. For example, you can see that this variant, so this is BA1, which means benign, stand alone. And with just this one evidence, this pathogenicity has been updated from not enough evidence to benign, which means with just this one piece of evidence, you already know that you can stamp the variant and say it's benign. But in most cases, this is not the case. So let's start with another case. So let's say now we have a variant, and we know that it's a truncating variant, which means with this variant, the gene may lose its function. So I say that and you add this. And now it says uncertain significance, which means you need more evidence to stamp this variant to pathogenic or not. Let's go back to something else. Say you have the segregation data, or you have allele data, or you have some other databases that you can play with. You can type all these different type of evidence here. But most likely, let's make this thing simple. Say we have another strong evidence saying that, let's go back to this. It says some amino acid changes as an established pathogenic variant. Okay, so let's say that it's the same as, let's just give another famous change here. We add it here, and now it says it's pathogenic, given the guidelines. So we can do a lot of things. For example, if you think that the evidence is not PVS1, it's not very strong, you can change it to strong, and see how the rules evaluates for you. In that case, because you have two strong evidence, then the system still consider that this variant is pathogenic. So by giving something like this, we are confident that we can give people a feeling about how they can adapt to this new guideline. And actually, this guideline have other unmentioned functionality as well. For example, you can share your curation work to other people. Say, I can share this work to Dr. Zhao and say, hey, Dr. Zhao, probably you are interested in this variant as well. How about you contribute your knowledge to this? Then we can just give Dr. Zhao's email and then send this thing to him. Okay, I think the system cannot recognize this, but for now this one is good. We can also say that, so for example, if you curate a lot of different types of evidence, say, and you really want to say that I think this one is very interesting, and you want to really share this work and save this work for later work, then you can actually download the data. You can download data as text, or you can download the text as JSON. So for whatever your purpose is. And once you download the data, you can actually keep working on the data when you have time later. And
such kind of record is individually kept.
Yes, it's in the database. Yes. For example, if you want to go back to this work later, you can just copy paste this one and go back to it later. You still have all this curation work saved from our database. So actually, in addition to implementing these guidelines, you actually make this curation process more like a crowdsourcing project, which means everyone with the knowledge can contribute to this. You don't have to ask me first, say, Shane, I want an account for this. No, you don't need anything. Just go to this website. You have a unique token at the top, which means only the person who has the link can access to the data you have. So it's very confidential. But at the same time, because this unique identity system, you can easily find your curation and share it with others, with your collaborators.
How much of this evidence can be populated automatically?
That's a good question. I think at least for this population frequency data, it can be populated. Yeah. And also for, I think for this part, for this some amino acids, because I think there is a database already, somehow what the database does is to have a list of simulative variants that are sitting in the same codon with some known pathogenic variants. So you can basically just go to that database and pop up that for this cell. And whenever you type, for example, in that very name, instead of giving a dummy name, you can just give, for example, RS. You can give a real variant identity. So that way the system can automatically look up the information in the background and fill up the cell for you whenever they can. But for many parts of I have to mention that for many parts of the cell, such as, for example, this truncating or the segregation analysis, you still have to do it manually. Because that part, I think there are no existing solutions that can do that for you automatically.
So out of the ones that are remaining, is it because the data isn't organized in the right way?
Exactly. That's one particular reason. And also, another big reason is many of the curation process, like you have to decide if the family is segregated with this particular gene, like PCSK9, you probably have to go to the pedigree and look up the pedigree for that particular thing. Then I think maybe the pedigree analysis part is automated, but that part, if you want to integrate that part with this system, you still have to write another piece of code to do the gapping work, the gluing work.
So is there anything in here that that can't be automated?
Yes.
For example, like if it requires some human intuition.
Exactly. I think for that, as I mentioned, I think that part is most likely in a segregation analysis. Yeah. But I can certainly try to think about like a better way to at least automate a part of that process so that people have a better efficiency. So that's the part. I mean, the part I was just talking about is about like how to populate the database from both consolidating the databases as well as enabling others to contribute to your database. And now we have some data in the database, then we can play with data by ourselves. So the first thing we did is actually just very simple. We just look up the database for a very famous gene called APC. It's actually related to colorectal cancer. And the question we want to ask, or want to answer, or want to address is, are there any particular locus in this gene, particularly in some exons, that are more frequently mutated than other locations? So because we have the data, what we can do is to actually just count the number of SNPs in a particular location. And actually after this simple process, and this is the overview of this APC gene, you can find that in the middle exons of this gene, we have a known mutation hotspot that has been reported previously. We also identify this mutation hotspot. We also identify some other new mutation hotspot that is not previously reported. And actually, if you zoom into this last one, the biggest exon, we also have two previously reported mutation hotspots validated just using this very simple counting method because we have the data. The data is the power. So, excuse me. Once we establish that part, we want to do some exploration. We want to do some like a guess about, are there any new mutation hotspots in more important genes such as BRCA1 and A2? Actually, we found that there are quite a few actually, especially for this big exon. I think there are at least 5 or something. This is the zoom out view, so I cannot provide details, but I think for that particular exon, it's actually quite interesting. We also did similar things for BRCA2. The result is actually quite similar. But I think this one is a little bit interesting here because if you think that the size is a bias for the chance to have a mutation hotspots, then this one is say no, because this one is almost the same size as this exon, but it only has 1 mutation hotspot. So it's quite interesting to see why this particular exon, because maybe it's located in a particular like protein structure or domain, so that is so important, so crucial for the function of this gene. So that's why the ClinGen project also considers machine learning algorithm based on protein structure data. The second thing we did is actually to try to empower the data we empower the persons, the curators, we also want to empower the data. And the way we empower the data is actually integrate the original variance data with some ontology, such as the HPO, the human phenotype ontology, or some other famous ontologies. Actually, the author of HPO, Peter Robinson, just published a study where he tried to automate the diagnostics of the genetic database diseases by combining the ontology inferring and a customized panel with disease-associated variance sequencing. So what he did is he used the ontology to automatically infer most likely diseases based on the patient's description, and then use that to somehow combine that with the variance sequence from a very tiny customized disease-associated panels to make sure that they get the most meaningful variance. Here, what we did is somehow in a similar way, but not that complicated, because what we have is some public data from some local specific database. In this case, we're playing with the gene TP53. Of course, everyone knows about this gene. So what we found is if we map the variance to this ontology map, just based on the patient's disease. For example, some patients have lung cancer, some patients have liver carcinoma, then we map them first to some leaves of the tree, and then we can, of course, intuitively infer their higher level leaves or their parents just based on, just relying on the ontology structure of this HPO or maybe ordo or something else. But in this case, because we're talking about cancer, probably HPO is better than ordo. So in this case, we can say that this variant is lung cancer related variance, but is also related to respiratory system. So in this case, we can separate the variants based on their different, annotations in the human ontology, and we do see that they have different distributions, at least in certain exons for this gene. I think this is just a starting point compared to what I mentioned previously, because I think this one, as mentioned, after I read Peter Robinson's paper, I think this is probably something we can further investigate. The last thing I want to show you is some, I would say, Crow's database variant identity analysis. This is what Dr. Zhao just asked. I think if people come from different databases, they have different requirements, and the first thing probably they want to ask is, are my variants in your database or not? And actually, this question becomes much more important for the developer of the database ClaimVar, excuse me, and also other local specific database. Why is this question so important? Because now the policy is ClaimVar wants these RSDBs, these different data silos in different labs to submit their data to ClaimVar. Then before they submit, they have to do some evaluation. Like, for example, if I have all the data you own in our database already, then probably I don't that much need your data submission at all. So in this case, we analyze two genes, BRCA1 and APC again. So in the case of BRCA1, we see that most of the data in the ROVD, which is the most famous local specific database for BRCA1, most of the data has been in ClaimVar already. But another very important database called UMD, which is another local specific database for BRCA1, only have about like half in ClaimVar. So by viewing this graph, Heidi Ram says that, oh, we really need to push UMD, the database in France, to submit their data into us because we really want their data. This is almost like you see, like 50% gain for the original database. And in the case of APC, the thing is, I think it's much easier. As you can see, most of the genes in ClaimVar are already in DBSNP, which is the truth, because whenever ClaimVar has like a new release or significant gains in terms of variance, then ClaimVar is required to submit their data to DBSNP. So it's expected that most of the data in ClaimVar are already in DBSNP. But we also found that compared to the case of BRCA1, which is a much more famous gene compared to HPC, the variance is relatively still sitting in different silos. So by viewing this graph, then we can make a decision that, okay, we need to somehow require more efforts, more help, more attention from the maintainer of these two databases so that they can push the data into our database. So this two graph is actually the most cited slides of all my work during all these consortium meetings, studies. So I just actually initially when I come out with the two graph, I don't think that really that too important because they are just used for our internal discussions. So I think overall our focus is on the data, as you can see already from my presentation. We want to somehow incorporate so that we can use the least amount of resources to somehow attract the biggest possible volume of data into our database, and somehow we use this centralized database to support the analysis of genomic variants, and somehow we can hope that we can bring this annotation-based or variant-based analysis into a better healthcare situations. Just like a paper, we also had talked about some limitation of this study. I think many of you probably have heard about the GA4GH, which is a global alliance for genetic health. Their goal is actually quite opposite to what we're trying to do. They're trying to say, okay, so everyone has the data, everyone has a silo, and not too many people want to share, so what do we do about this? So instead of asking, for example, ask the people in UMD or LVD to submit the data into CleanVar, okay, you don't have to do this. Then instead of submitting data directly to our database, what you can do is to follow certain process and make sure that your data is publicly shareable, accessible, or even writable by every other people who have this token. So it's more like a federated system. And our system is more like a wheel hub design. Like you have this hub, and everyone comes into this hub through the wheel or something like that. So I think so far, the CleanVar is still very successful, but GA4GH is also getting a lot of attention and traction. So it's really hard to say which policy is better than that. So we probably have to see. But our goal is that we hope that by our efforts, we hope that in a few years later, when we come back to this slide, I can see that we were getting really a lot of knowledge and actionabilities into the things, into the black hole, which we once knew little about. So I think that's my talk today, and thank you for your attention.